retrospectively go in and select samples that we're interested in. For example, based on to take samples from children that we know develop certain diseases later in life. So this is something that we have utilized in this study to study autism. So autism, or autism spectrum disorder, is characterized by communication difficulties, repetitive behavior, social impairments, etc.
There's an issue with the screen sharing person.
Too bad. Yeah. All right. And the incidence rates of autism is on the rise in many regions of the world. Some places reaching over 2%, actually. Although there are several risk factors that are known for autism, such as genetic factors, but also early life exposure and prenatal exposure, as well as being male, which is associated with a very large increased risk. The overall molecular mechanisms are more or less unknown. So this is where we want to bring in the neonatal metabolome in order to kind of map the early metabolomic alterations that we can see in children that later develop autism, and use this to better understand the pathophysiology of autism, but also potentially, at least in the future, to facilitate earlier and more accurate diagnosis of this very heterogeneous disease. So that's why we designed this study using samples from the Danish National Biobank. So we used samples from children that were born between 2003 and 2008. So, and then we defined autism as children that were diagnosed with autism before between the age 1 and 10 of their life until latest 2015. We also looked up samples from children that were that we matched for these cases based on factors that we know are very important for the neonatal metabolome, including the age of sampling, the stational age, and the date of birth. So in total, we analyzed 700 or so cases, each with a matched control, which ended up being then close to 1,500 samples that we analyzed using our LC-QTOF system. We use data-dependent acquisition, which you already heard about this morning. Our pre-processing was done using MZ-MIND3, which you will hear all about tomorrow in detail. This ended up being measuring 865 metabolite features that passed our QC procedures, our filters. These features were then annotated using GMPS and Sirius, which you will hear about on Wednesday in more detail, resulting in 200 or so, a bit more, 229 annotated metabolites, while the rest remain unknown in the dataset. So if we look a bit at the results, we saw in total, we found almost 100 metabolites that were significantly associated with development of autism later in life, using a nominal p-value threshold. These included, and we analyzed these using logistic regression models, where we also adjusted for factors. These factors, I already said, are important for the neonatal metabolome. If we just look a bit at some trends in what metabolites actually popped out here, we saw several of these acyl carnitines, you can see here. So this is C2 carnitine, for example. We also saw quite a bunch of metabolites that are produced by the gut microbiome. For example, these indoles that come from the tryptophan metabolism produced by gut microbiome. So the majority of these metabolites were associated with an increased risk of autism. However, when we adjusted for running very many different multiple tests, which you will hear more about as well during the stats section on Friday, only 2 metabolites remain significant, and those were cycloleucine proline and 5-aminovaleric acid, or in detail, just at these two metabolites. And we stratified our analysis after the age of diagnosis. So we set a threshold at age of 6. So then we saw that 5-AVAB had a much stronger association with autism for kids that were diagnosed early in life, whereas we could not see a significant effect for those that were diagnosed later. All right, so of course you want to know a bit more what these metabolites actually are. So cycloleucine-proline is, as you can tell from its name, a cyclic dipeptide. And it's interesting because these molecules are known to be able to cross the blood-brain barrier. So it kind of makes sense in this context that they are known to be present in the brain. We also know from previous genome-wide association studies that variants in these two cytochrome P450 genes, so CYP1A, 1A2, and CYP2C19 are related to the levels of these metabolites in the circulation. So partly just a bit curious, I think that this gene, as some of you may know, is the gene that is responsible for degradation of caffeine, which is also a bit interesting because we saw in our data set that metabolites that are downstream from caffeine in the caffeine metabolites were quite strongly correlated with our metabolite of interest here, which I find a bit curious. We also know that CYP2C19 is responsible for degradation of several of these antidepressants. Okay, so 5-AVAB is structurally very similar to a carnitine, and it's been known from studies in mice that it's present in the fetal brain. However, when the maternal gut microbiome is absent, so are the levels of 5-AVAB in the brain, which indicates that at least partly this metabolite is most likely produced by the gut microbiome, at least in mice. And then if we look locally in the brain, it's also been shown in this study in Nature a few years ago that in germ-free mice, if you supplement them with this metabolite, you actually stimulate the production of new axons. So it seems like this metabolite has some kind of effect locally in the brain, although we don't know exactly what it is, of course. So it's something for the future, for sure, to look more at. Another piece of the puzzle was brought to us by some of my colleagues and our collaborators, where they looked at the neonatal metabolome and the maternal metabolome to see which metabolites were correlating between them. And then they saw that 5-AVAB, which is here, has a slightly different name. This is typically in metabolomics. But anyways, it's among the metabolites that was the most strongly correlated between the mothers and the child. To me, this indicates that what we see as we actually measure in the neonates is something that they actually get from the mother. So if that is true, it would be very interesting to know what can do to influence the levels of this metabolite in the mothers. So in order to answer that question, we contacted our collaborators in Sweden, and we got access to these two cohorts with untargeted metabolomics data. And these cohorts are good because they are very, very well phenotyped. So we have untargeted metabolomics data available, as well as dietary assessment data and genome-wide genotyping. So we looked into the untargeted metabolomics data to see if we can find a match between the metabolite that we measured in our data set that we annotated as 5-AVAB with the untargeted data that they have. And we use GMPS to do this, which you will also hear more about later this week. And we found a good match with a cosine score of 0.81 to 0.83 in the two different cohorts. So we assume that this is 5-AVAB, and I will refer to it as such in the rest of this presentation. All right, so now we wanted to know, can the dietary intake and the genetic predisposition explain the levels of this metabolite in the circulation. So we started to look at the habitual dietary intake. So if we look into both the cohorts, so each line here is a cohort, so it's MOS and MDC is each a cohort. We saw that increased intake of dairy was correlated with higher levels of 5-ADAP in the circulation. And this was true in both the cohorts, it's quite reproducible. So if you broke this down into the broke dairy down into its components. So for example, milk, yogurt, cream, cheese, and butter. We saw the similar trends across the board. However, it seems like milk is the driving factor here. Okay, so it seems to have something to do with dairy and diet in general. What about your genetic predisposition? So we performed a genome-wide association study and found a very strong hit for this gene, SLC22A5, which codes for an enzyme. Sorry, not an enzyme, a transporter protein that is responsible for transporting carnitine and also 5-ADAP into the cell. So both into the cell, but also across the blood-brain barrier is known from previous studies. And it's also been shown in previous studies that that having high levels of 5-ADAP in the circulation will decrease your cellular uptake of carnitine, which is very interesting. Also, it's known that 5-ADAP is can inhibit the biosynthesis of carnitine. So in the rate-limiting step, the production of carnitine in your body, 5-ADAP can actually inhibit this enzyme. So it has two separate effects on the production of carnitine, both the production and the uptake of carnitine into your cells. So what happens if you have low carnitine in your cell? Well, the result is that your carnitine is responsible for transporting fatty acid chains across the mitochondrial membrane. And if you have lower levels of carnitine, then your fatty acid metabolism, your beta oxidation will be affected, will be lowered. Which is interesting because it's been seen in several studies before that autism is related to changes in the in fatty acid metabolism. Also that carnitine, inborn errors of carnitine metabolism also has been associated with autism. So there's a lot of interesting things going on here. We try to put this all together to see if we can understand it a bit better. So just to conclude, we found that 5-ADAP and cycloleucine proline are increased in newborns who later develop autism. We saw that 5-ADAP could influence the risk of autism by modulating brain carnitine availability. And we think that if we can find a causative link between 5-ADAP and autism, it can be targeted in dietary or potentially also pharmacological interventions in the future. So with that, I want to conclude and by saying big thanks to everyone in the Metabolomics group at SSI, especially to our group leader, Madeleine, which have been also very involved in this study. They all have. And thanks to you for listening. And I ended up just 2 minutes late, so it's pretty good, right? Thanks. I think if you have any questions, I don't know if we should do it now or if just maybe you can just approach me afterwards if you're interested and we can have a talk. I just wanted to say a few practical things. So for tomorrow, we have you can actually get already now your the kind of access cards you need to get in that you can get down from Adam down there. The thing is that what you need to do when you come in the morning is to ring the bell and the reception will let you in, but you don't have to go into the reception to register. So when they let you in, you can just go straight here. But remember to bring the access card because yeah, it's mandatory. So so that's it. And then we will have the social event with the food truck coming up soon. So let's meet up there.